Good morning or good afternoon to everyone, depending on where you are joining us from. And welcome to today's webinar on the rights to use CRISPR-based tools. I'm Alessia, and I'm the responsible of the scientific communication at genOway. And I'll be the moderator for today's webinar hosted by genOway. And now let's give a chance to all the speakers to show their smiling faces. So please, Amélie, Sandrine, Mathieu, hi. Amélie, hi. Ajay, hi. Okay, thank you so much. So the aims and objectives for today are that you go away understanding more about CRISPR-based tools and the patent rights you need in order to generate rodents, animals, and cells. Before we start, a little bit of housekeeping. So today's webinar is going to be 30 minutes long, and we will have a chance to answer to your questions at the end of our webinar. There are a number of ways you can use to interact with us during the webinar. For example, if you use Twitter, you can join in the conversation using the hashtag genOway events. We will also have a poll question during the webinar that will pop up right on your screen. And finally, you can send your questions ready for the Q&A session using the button questions. We now come to today's poll questions. So as you can see, this is the question. What is the main reason why you are participating to this conference? And we have 5 options. You are considering developing rodent models using CRISPR. You are considering using rodent models using CRISPR. You use rodent models and wonder which patent right you need. You want to learn more about CRISPR technology patents. And finally, you want to learn more about how genOway is using CRISPR. This is a multiple choice poll, so you can click more than one answer. So please make your selection, and then we will share our results. Actually, I see that most of you voted. So we'll wait a couple of seconds more, and then so now we can show the results. So I see that 6% of you are considering developing rodent models using CRISPR. 8% are considering using rodent models using CRISPR. 14% of you already use rodent models and wonder which patent rights you need. 88% of you want to learn more about CRISPR technology patents. And finally, 33% of you want to learn more about how genOway is using CRISPR. So thank you so much for your answers, and we'll try to answer to all your questions during our webinar. So now it's time to introduce our first speaker of today's webinar, Amélie Rezza. So Amélie received her PhD in molecular and cellular biology in 2009, and then she moved to the Icahn School of Medicine at Mount Sinai in New York as a postdoctoral fellow. She authored 6 first author papers, including her last one on CRISPR-mediated gene editing that was recently published on Scientific Reports. And in January 2017, she joined genOway as Innovation Project Manager and Cell Specialist. Today, Amélie is going to tell us about the use of CRISPR for model generation.
Thank you, Alessia. Yes, so I'd like to give you a very short introduction about CRISPR, because it has really been a revolution in the genome editing world. If we take a timeline of the different gene editing tools that are used and that have been used, the first one to have been widely used is the homologous recombination. It was first used in the mid-1980s. Then came along the programmable nucleases. The first one to be used were meganucleases in the mid-90s. Then zinc finger nucleases in the early 2000s, TALENs in the late 2000s, And then last but certainly not least, our suspect of interest today, CRISPR. It was first used as a gene editing tool in vitro in 2012 and in vivo in 2013. The way programmable nucleases work, and this is the reason why they mostly took over homologous recombination, is that they can boost gene editing frequency and efficiency. So here's a schematic of how it works. These nucleases can induce a double-strand break in the DNA at a specific sequence, and then it can be repaired in one of two ways by the cell. Either there is random reparation with insertion or deletion of a small number of nucleotides. These are called indels. Or if you provide a donor DNA to the cell, it can integrate this DNA precisely at the cut site, and that's what we call precise integration. Different type of models can be obtained with these two repair mechanisms. Either a knockout by frame shift with a random reparation, but with using precise integration, you can have a lot more different models. So you can have a knockout by the insertion of a stop codon, or every kind of knock-in, point mutant, reporter, humanization, really any kind of knock-in you can think of. In the gene editing community, we mostly use, pretty much always use precise integration because it's really more reliable and we can predict the outcome of the gene editing, which we cannot do with random reparation because it's random, obviously. So CRISPR has been a revolution, really. And the reason why is that it appeared easier than the previous programmable nucleases that existed. As an example, for zinc finger nucleases and TALEN, for them to work, you need two custom-made fusion proteins, and the specificity of the targeting in that case is controlled by these fusion proteins. For CRISPR, on the other hand, also you need two partners. One is a protein, for example, Cas9, always the same protein, and then one custom-made RNA molecule. In that case, the specificity of the targeting is controlled by the RNA molecule, meaning that if you want to target a new locus, when using the zinc finger nucleases and TALEN, you need to custom make two fusion proteins, whereas in CRISPR, you only need to make a new RNA molecule. Of course, RNA molecules are easier, faster, and cheaper to make, which is why CRISPR became so popular so fast. Indeed, if you look at the number of publications on PubMed per year, here you have in black TALEN publication. They kind of plateaued around 2015, around 250 or 300 publications a year. But if you look at CRISPR here in orange, it's actually growing exponentially, and it's not finished yet. So here at genOway, we try and want to contribute to the development of gene editing tools. We've been working for 20 years now in the generation of reliable, relevant, genetically engineered rodent models, specifically for preclinical research. If we take this timeline again, we have here, so genOway was created in 1999, focusing mostly at the beginning on mouse models. Then in 2003, we developed the first technology to clone rats. And we had since have many R&D programs focusing on nucleases starting 2010. And since 2014, we really focused on the optimization of CRISPR technology. Indeed, there are still some things to be optimized about CRISPR because there are some limitations to its efficiency. If we take this schematic again of the two ways to repair the DSB induced by CRISPR, the two main ones that are focusing the most interest in the gene editing community are off-targets. These off-targets are due to the fact that CRISPR can bind to unwanted sequence, untargeted sequence, and in that case, if CRISPR binds, it cuts, and if it cuts, there is a repair, either by random reparation or integration if there's something to integrate. And in that case, these are off-targets. We want to avoid this, obviously. The other limitation that we are particularly interested in genOway, and we're really working very hard on this, is the fact that the efficiency of precise integration for large knock-ins is still pretty low with CRISPR. So we are really focusing on these on-target events here at genOway. So as a summary, Clearly, CRISPR-mediated integration is absolutely the most efficient gene editing tool available today. There are still remaining limitations that a lot of laboratories are working on as of today to lower these off-targets I just mentioned, and increase large knock-in efficiency. With these optimizations, we will be able to best predict the outcomes of CRISPR gene editing, which will allow us to increase its efficiency and, most importantly, its safety. So it's interesting to see that the next technical step for CRISPR are pretty clear, which might not seem to be the case for its IP landscape. And this is one of the reasons we wanted to have this webinar today. So I thank you for your attention and hope you have some questions.
Thanks a lot for your amazing presentation, Amélie. And now I'm going to hand over to Mathieu Fontaine. So Mathieu obtained his master's degree in biotechnology, proteomics, and genomics at the University of Toulouse in 2010. Then he moved to Belgium, where he worked as a research officer in the field of oncology. In 2013, he attended the Grenoble Business School, where he developed skills relevant to the biotechnology industry and intellectual property management. In 2014, Mathieu joined Questel company, where he is now Senior Consultant Life Sciences, specialized in biotechnology and health. Today, Mathieu is going to give us an overview of CRISPR patent landscape.
Thank you, Alessia, for this introduction. As you mentioned, I'm now going to give you an overview of the highly trendy CRISPR technology from a patent perspective. Next, please. Indeed, as you can see on the first graph, this field has experienced an important increase in interest since the filings of the 5 key patent families in 2012, with currently around 3,300 filed patent families. Some works were protected before this date, but this is truly the combination of the CRISPR enzyme and the Cas9 protein that made this field so appealing, thanks to the ease of use and the precise targeting of DNA loci. As this technology is in fashion nowadays, more and more patents claim the use of CRISPR in extensive lists of molecular tools, such as zinc finger nucleases or TALENs. Regarding the top players, the cluster MIT, Broad Institute, Harvard, Editas Medicine leads the race with the biggest portfolio size. Zhengfeng is a key inventor of the MIT, and several others from this institute are also recognized as key inventors in the field. The UC Berkeley, under the affiliation University of California here, and which corresponds to the work of Dr. Doudna, took also a strong position in the field with around 100 patent families. Remaining players are mainly academics, but some industrial players are also present in the top 20. DuPont, Sangamo, CRISPR Therapeutics, and Cellectis. Overall, US and Chinese players are leading the field. Next, please. Reflecting the top players' nationalities, the main targeted markets are China and the US. Europe is lagging behind with 2 times fewer patents filed in this region than in Northern America or Asia. Regarding key R&D locations, the US are the main engine, followed closely by China. Other countries contributed far less than these 2. Next, please. Even if it is a young field, plenty of areas of investigation have been explored. On the island graph presented here, you can see plenty of small islands corresponding to clusters of innovation. We grouped clusters according to the 5 main areas of investigation. First, CRISPR tools, mainly enzymes, RNAs, and reagents to perform gene editing. Second, the therapeutic or diagnostics approaches for humans. Due to its increasing incidence, oncology is the main area of development, especially via the CAR T-cell approach and immune cell engineering. Blood-related diseases attracted also a lot of interest, with gene therapies aiming at replacing the deficient factor VIII gene. Material delivery thanks to viral vectors, and that is to say lentiviruses or AAVs, have also been extensively explored. Thirdly, cell engineering. Whatever the final goal, mammalian, bacterial, and yeast cells are modified thanks to CRISPR. The fourth category, plant engineering, with a strong emphasis on maize, corn, and rice-modified crops. And lastly, animal models. Knockout models mainly developed, but we see also the emergence of knock-in approaches. Next, please. By focusing on the CRISPR-Cas9 technology, which represents 60% of the patents of the whole landscape, interest here is even higher than in the whole field, with more than 70% of annual growth. Nonetheless, top players, main targeted markets, and R&D locations remain the same as for the whole field. Next, please. And now for this last slide, we focused on the year 2012. We can identify the 5 key patent families that triggered interest in the CRISPR-Cas9 technology. They are owned by 5 different players, and the global extension strategy has been preferred by all these assignees, as you can see on the slide. The University of Vilnius filed in first on the 20th of March. The UC Berkeley followed on the 25th of May. Toolgene, a Korean company, filed on the 23rd of October. And finally, Merck and the Broad Institute filed both on December the 6th and the 12th, respectively. Only 2 of these patent families are famous due to the ongoing high-stake legal battle between the UC Berkeley and the Broad Institute, but the 3 others are also key and considered in the litigation. Content of these patent families will be detailed and compared in the following presentation for you to understand what they cover exactly. Thank you for your attention.
Thanks a lot for your excellent presentation, Mathieu. And now our next speaker is Ajay Rastogi. Ajay has a technology and business background with over 20 years of worldwide licensing experience in the pharmaceutical industry. In his present role, he is Director of Business Development and Licensing and Gene Editing and Novel Modalities at Merck. In December 2019, he played a key role in negotiating and execution of the company's first exclusive CRISPR integration license for rodent models with genOway. Today, Ajay is going to tell us about the coverage of specific Merck CRISPR-enabling process.
Thank you, Alicia, for the kind introduction. I'm Ajay Rastogi, and I'm with Life Science Division of Merck. I'm very pleased to provide an overview of our foundational integration CRISPR intellectual property. An important reason for our participation in this webinar today is, as Alicia mentioned, we have provided genOway an exclusive sub-licensable license to develop rodent models and to edit rodent cell lines for internal and commercial use. Next slide, please. As you can see from the slide here, we have been a pioneer and innovator in gene editing technologies and have been a world leader in offering commercial products for gene editing, starting way back with Targetron in 2005. Then in 2008, we obtained an exclusive license from Sangamo for use of zinc finger nuclease technology for research use with rights to sublicense. And in 2013 onwards, we have been active in developing, patenting, and commercializing CRISPR technology and products, and have received coveted international awards and recognition. Next slide, please. Here we show how our foundational patented CRISPR integration technology enables cutting of the DNA using guide Cas9 that enables highly targeted insertion. Next slide, please. Here is an excerpt from our granted European patent, and I would like to emphasize the critical aspects here. Number one, this patent enables insertion of exogenous nucleic acid sequence. It applies to all eukaryotic cells. It covers in vivo, ex vivo, and in vitro use. It includes CRISPR-Cas type 2 systems. It covers a double-stranded break in the DNA. Next slide, please. We have also applied for patents on paired nicking that greatly reduces off-target activity and increases specificity. Next slide, please. So what is unique about our intellectual property? This IP is essential for use of any CRISPR-Cas9 gene editing for insertion or knock-in. We have the only granted IP for knock-in, and we have constructed a robust portfolio of 14 granted or allowed patents in 8 countries, and this number is increasing almost every month. The follow-on patents serve to enhance gene editing using CRISPR by improving accuracy, specificity, and reducing off-target effects. Finally, we have been very successful in offering exclusive as well as non-exclusive licenses, and we welcome all licensing inquiries from all attendees today. Next slide, please. This slide depicts the areas where we are offering our CRISPR technology for license. So the areas are basic and commercial research, for example, cell models. This could be tools, reagents. Users could be CROs, for example. The rodent model licenses are available from genOway. Other areas include industrial and ag bio, for example, crop traits. Therapeutics is a very important application for CRISPR, as Matthew earlier alluded. And here, CRISPR-based gene editing research is aiming for cures in hemophilia, sickle cell anemia, thalassemia, CAR T therapies covering oncology, and cell therapies. Another area of active interest is diagnostics, food and beverage. There are a number of clean meat companies looking at CRISPR, for example, and animal health. Next slide, please. Here you see the 3 buckets in which we compartmentalize our CRISPR patents. So these 3 buckets, number 1 is our foundational CRISPR patents covering insertion. The next bucket in blue are follow-on patents like proxy CRISPR, CRISPR Chrome for enhancing specificity. The third bucket in yellow are paired nicases and proxy CRISPR again, which reduce off-target effects. And the takeaway message from here is that when we offer licensing rights, we offer all 3 buckets as a single license. Next slide, please. Our CRISPR portfolio status is shown here. And what we are showing here is a roadmap of the different patent buckets, showing where and in which countries patents have been granted and where they're pending. Next slide, please. Here is a table in greater detail with patent numbers, and interested viewers may contact me later for more details. Next slide, please. This is a depiction of a geographical roadmap that emphasizes how widely Merck is committed to patenting globally. As you all can see, our patents have been either received or pending or granted in USA, Canada, Brazil, Europe, Israel, India, China, Singapore, South Korea, Japan, and Australia. Next slide, please. And finally, I'd like to share some recent developments in news as far as our relationship with genOway is concerned. Next slide, please. And this is the press release of our exclusive license to genOway for rodent models, recently executed in December 2018. And through this license, genOway has the rights to sublicense use of our CRISPR for developing rodent models and also for offering edited cell lines for internal research and commercial use. Next slide, please. So as mentioned, we would be very happy to discuss licensing. Please contact me for entering into a CDA in case of an interest in any non-rodent field, and my contact information appears on this slide. Thank you very much.
Thanks a lot for your amazing presentation, Ajay. So now it's time to pass the voice to our last speaker of today's webinar, Sandrine Carteau. Sandrine received her PhD in oncology in 1993, and then she moved to the Salk Institute in San Diego, California, as a postdoctoral fellow. She then worked in several biotech companies as a researcher and business developer, and then at the University of Lyon as technology transfer officer. In 2012, she joined genOway as intellectual property and licensing department manager. Today, Sandrine is going to tell us about the freedom to use CRISPR-Cas9-based rodent cell and animals.
Thank you very much, Alicia, for this kind introduction. So let me present you what we can do for you. So the question I'm going to ask is, do you have the right to create and use CRISPR-based rodent cells and animals? Yeah, I know it's a tricky question. And for me, working with CRISPR is like entering into a battlefield and trying to find a safe way out. But I'm going to show you that there is a safe way out in the rodent field. So let's go back to the applications with rodent cells. And when I say rodent cells and animals, what Ajay called the rodent models. So there are many applications you can do with this, and you can rely on the rodent models. So as an example, one could do a target validation assay. And so for this, you are going to knock out your gene by inserting a stop codon or by creating a frame shift into the gene. And then you go, if you want to go further in the study, you can develop a humanized, what we call a humanized model. So it means that you insert the whole cDNA of the human gene or the human cDNA of the human gene or the human gene. And as you can see, many of these models are knock-in models, meaning that you need to insert an exogenous DNA into the genome. And the CRISPR technology has been a revolution in the gene editing. And thanks to Dr. Chen and Dr. Davis from Merck, who have invented the enablement of this insertion by CRISPR, it's the insertion and the development of knock-in has been very easy. But now, how to safely create and use a CRISPR-based model? So let me draw a mouse with slices. Each slice corresponds to a patent portfolio. If you want to work safely with your model, you need to have a license on all the patent portfolio, because if you have all the licenses but one, in that case you don't have the freedom to operate. You are not going to work safely with your model. And the other licenses can be from Vilnius, Berkeley, Tulgin, Broad, and many others. And as Mathieu said, there are plenty of different patent portfolios. So no need to say that I can't tell you now which patent portfolio covers your model. But what I can tell you is that if you created your model by inserting a sequence into the genome into the genome, the fact is your model is covered by the Merck's patent portfolio. So how can genOway help you with these models? And as Ajay said, we acquired an exclusive right to Merck's foundational CRISPR patents. So, and we do have a sub-licensable exclusive license under the patent rights to make, use, breed, and sell rodent cells and rodent animals. And we do have all the non-exclusive rights under the other needed patents rights covering the models developed by genOway, meaning that we do have all the rights that to transfer you the model with the freedom to operate, and we guarantee this freedom to operate. We have acquired this exclusive license. My goal is to make these tools available for most of the researchers. And we, from our 20 years experience, we think we know a little bit what you need. And so I'm going to show you our solutions for you. And because the non-profit organization and for-profit organization don't have the same needs, I'm going to start with the non-profit. So we can offer you the freedom to operate for all the models we develop for you. And if your co-facility develop the model, we can offer you the rights under the foundational Merck's patents, because we do have the exclusive sub-license. So we can offer you the internal R&D, the use for internal R&D purposes, so for free. And we also offer you for free to work with third party and for third party for an academic laboratory. I think it's very important to be able to offer third-party services to companies. And if your model was created by your core facility, we can have you grant you the rights to work with for-profit companies under an annual flat fee license. So if you create model for third parties, we can help you to distribute this model to external academic institutions. And last but not least, we can remunerate you for the distribution of the models and that you have developed or that genOway has developed for you. And we can remunerate you for the as a third-party intermediate if you transfer a request from a company. So for biotech and CRO and pharma companies, we know that you need to do internal research and to work with third parties. So we can grant you and we are going to grant you all these rights, and along with the freedom to operate with the model we developed for you, and under a one-time flat fee per model for worldwide use and for an unlimited duration, even if you sourced the model from a third party. So, and you will be able to do commercial applications under annual fee if genOway develops the model for you. It sounds the same if a third party develop the model, but just remember that we can only offer you a license on a specific patents, because we can't transfer you the freedom to operate in that case. So just to let you know that our valuation, the valuation of our licenses, takes into account the current complex CRISPR IP landscape. And so it means that the pricing of our license are very adapted to your needs and this landscape. So the take-home message of my presentation is that genOway is the only party able to provide you with a CRISPR integration-based rodent cells or rodent animals, and along with the guaranteed freedom to operate to secure your project. Our offers are customized to fit your and your partner's needs. And so I have not the time to go through into details on the other offers, but I would be very happy to discuss with you and to present these offers in more details under CDA later on. Thank you very much for your attention.
Thanks a lot, Sandrine, for your great presentation. And now is Q&A time. So if you want to ask a question, please do so by clicking on the button questions, and we'll try to answer as many questions as we can. So we already received a few questions. For example, we have a question for Mathieu, which is the following. You have identified that knockout animal models seem to have been extensively studied. What about knock-in animal models?
For the moment, as Amélie said, there is problems with knock-in efficiency and off-target problems. So there is not so many patents compared to knockout models, because the CRISPR technology brought a good advantage compared to other technology, and especially knockout by frameshift. So for the moment, we see this emerging, but there are not so many patents on this matter.
Okay. Thanks a lot, Mathieu. Thanks a lot. So I have another question. This is for Sandrine. So in case an academic lab has created a model that he wants to distribute, how can you help?
We can... Sorry for the microphone. We can help in if we do have all the licenses that cover the model. So we can analyze all the techniques that were used to develop the model, and if we have all the licenses that cover these models, we can acquire this model and transfer it as a distributor to the for-profit with the freedom to operate.
Okay, thanks a lot, Sandrine. Now I have a question for Ajay. What is Merck licensing model for the integration IP and how this align with genOway?
So that's a good question. Merck independently is licensing, offering for license our CRISPR foundational IP for non-rodent applications. So as I indicated earlier, therapeutics is a key area where we are focusing, and Big Pharma, for example, is starting out taking a license for internal research. There are companies that are in the tools reagents area that have an interest in making edited cell lines. So we are dealing with them directly. Another key area is agriculture, where we have had a lot of success working with companies that are very basically ingrained in providing research, providing development work in the ag bio area. So we are offering licenses there. The other area where we have an alignment with genOway is in rodent models. So anything related to developing rodent models, genOway has worldwide rights to sub-license and also provide a CRISPR technology for editing cell lines, rodent cell lines. So those are areas where genOway has the lead. And we work jointly in many areas. For example, if there are pharma companies that are interested in rodent models, they would work with genOway, and we would work with them for other applications other than rodent models.
Okay, Ajay, thanks a lot for your answer. And now I have another question for Mathieu. The field knew an important growth until 2016. What about after?
For the moment, it's difficult to conclude because there is the 18-month delay in publication of patents. So I would say that it's going to increase again as the same way as Amélie showed for the publications. So I would say that the field is going to exponentially expand. Sorry.
Thanks a lot, Mathieu. Now I have a question for Ajay, which says, what happens if the ownership of new varieties of plants that have been edited with CRISPR?
So our licensing model, the way we are working with companies, if it is for internal research, and the company then develops a very specific trait, they can then convert this license, internal research license, as an exclusive license or non-exclusive, if it is available at that time, for application to a specific trait or a crop. So we would have to assess the situation at that time. If the license is still open and the licensee wishes to take an exclusive license, we would be happy to negotiate an exclusive license in that case.
Okay, great. Thanks a lot for your answer, Ajay. Now I have another question but this time is for Amélie. So how can you boost precise integration over random reparation after CRISPR cuts?
Okay, that's a very good question. Actually, a lot of people have been working on this, are still working on this. And here at genOway, we really, it's a main focus of our R&D department. Historically, there have been efforts to, for example, add chemicals and try to push the system towards this precise integration with the donor DNA towards towards this integration rather than the random indel apparition, the reparation. It's still an open question. Unfortunately, I don't have a clear-cut answer to that because a lot of people are still working on this, but hopefully soon we'll have something that will really increase efficiency of the knock-ins.
Okay, thanks a lot, Amélie, for your answer. Now we have another question, which is for Sandrine. For academic institutions, do they need to license from genOway in order to create mouse models?
You don't need a license from genOway, or you will have a free license from genOway to create a model in your institution for your own use. For example, if your co-facility creates a model for your university, we can offer you a free license. If your co-facility wants to create a model for an academic institutions, but external academic institutions, you will need a license from genOway. And as I showed you, it's very we have very straightforward the offer if we create mouse models for you also. So I would be happy to discuss with you further on this topic. Thank you.
Thanks a lot, Sandrine. Now I have another question for Ajay, which is, in case we require to conduct knock-in through CRISPR-mediated gene editing, what licensing pathway do we need?
So what our objective here is to make the technology widely available, and I would welcome you to execute a CDA with us. And the next step is very simple. Once a CDA is in place and we understand your objectives, where you're going to be applying the technology, we will then provide you with a term sheet and we can discuss a license.
Thanks a lot, Ajay, for your answer. So we welcome other questions through the question button. Actually, we just received another question, and this is again for Ajay. So the question is, in the case of developing a new variety of plants, fructal, for example, in a university laboratory using commercially purchased CRISPR, how to know what license you have?
So that is a good question. It's also a tricky question. We have to understand what your objectives are. Generally, for academic use, if it is only for internal research and you're not going to be earning any commercially sourced revenue, we are open to providing a free license. And all we request is that if you publish any papers based on your research, you provide us due acknowledgement. However, if there is a commercial aspect involved and you're purchasing reagents from a third party, then we have to be careful about who you're selling, and we would like to be very clear about the label license that you are responsible for when you're using those reagents.
Okay, thanks a lot, Ajay, for your answer. I have another question for Sandrine. Does genOway has an exclusive license for rodent cell gene editing?
Yes, we do have. Okay, so we do have. genOway has an exclusive license on the to create, use, and sell rodent animals and rodent cells. So it means that we're genOway the only company able to work with CRISPR and rodent cells, and at least unless you have a license from genOway, and we would be happy to discuss with you if you are interested in working and using CRISPR integration in rodent cells.
Okay, thanks a lot, Sandrine, for your answer. So please send us your question or comments via the question panel. And so yes, we have another one coming in. For knock-in applications of CRISPR-Cas9, are any third-party IP rights, for example, those of the Broad or Berkeley, required to be licensed other than the IP rights offered by Merck or genOway? I suppose this is for you, Sandrine.
Yeah, in fact, as I said during my presentation, you may need, and you may need the a license from the Broad or from Berkeley, because they are also foundational patents. So depending on the exact components you use and the exact way you use to create your model, and you use CRISPR system to create a model, you will, or you may depend on the Broad or Berkeley patents, of course. And we do have all the, as I said, we do have all the patent rights in order to develop and to transfer these models to a third party.
Okay, Sandrine, thanks a lot for your answer. We have another one that just came in. Isn't your exclusive license not only relevant regarding Merck IP? I suppose this is again for you, Sandrine.
But in fact, okay, I'm not sure I understand the question, that Daniela. The fact is, in fact, if you create a cell, a rodent cell line, and so if you create this cell line, and or if genOway creates this cell line for you, this cell line can be covered by the Merck's patent, but also covered by Berkeley's or by the Broad Institute's patents, or by the Vilnius patents. The fact is, we at genOway, we do have a person dedicated into the CRISPR patent analysis, and we do have all the licenses in order to develop these models. So that's our licenses. We do have non-exclusive licenses, and we do have exclusive licenses. So when we say licenses, they are relevant to Merck IP and or other licenses we do have.
And thanks a lot for your answer. And now I have another question. So did you say that the Broad and UC Berkeley licenses might not be needed to make a CRISPR mouse?
No, I'm not saying that. In fact, it depends. I'm just saying that it depends on the way you develop your model. So I can't tell you that if you need or not the UC Berkeley or Broad licenses to develop or to use a CRISPR mouse. So it really depends on the way you on the way you use the CRISPR technology. So, but it may yeah, you may have some ways where you don't need a license from Berkeley, or some ways where you don't need a license from Broad. First, depending on the country you are, and you are located, and also on the components you use.
Okay, thanks a lot, Sandrine, for your answer. So now this is the end of our webinar. So please look for our follow-up email that will alert you to the on-demand status of today's events, that you can review any material that you missed or simply go over again. So you will have access to the recording. Should you have any additional questions, please contact carteau@genOway.com. And then on behalf of Amélie, if you want to show your face, Amélie, Mathieu, Ajay, and Sandrine, as well as the teams at Merck, Questel, and genOway, thank you for joining us and taking time to view our presentations. Have a great day. Bye-bye.
Thank you. Bye.
Bye.